for not being physically present. And I would like to thank the organizers of SMIA for giving me this opportunity to present my research using this video option. So I'll be talking about what I call this integrated approach to the reconstruction of past human lifeways. Of course, we know that the end goal of historical and archaeological research is to create models that describe the society, the economy, and the culture of past human populations. And of course, these are reflected in aspects of past human lifeways, namely modes of subsistence and mobility patterns, and of course how these evolve in time. So chronology is also key here. Now, I propose to you that these aspects can be quantified through the use of Bayesian models. And one of the advantages of these Bayesian models is that they allow for the integration of diverse sources of historical and archaeological data. The starting point, however, for this approach is the use of multiple isotopic proxies for the analysis of organic remains. And here I'm going to emphasize the analysis of human remains and quantifying aspects related with diet and mobility. And of course, that implies that we need to know how the isotopic signals in the environment and in food are transferred towards these human consumers. And for that, we need data from feeding experiments. Now, these past human lifeways are framed within environmental and climatic histories, and these are defined through a series of both isotopic and non-isotopic paleorecords. Of course, it would be highly advantageous that all of this isotopic data is centralized in some way. And so I will be briefly talking to you also about the ISOMEMO initiative. However, as I said, the starting point for this approach is isotopic analysis. And here I want to emphasize the importance of using multiple proxies. And this is exemplified in this rather simple model. Now, this simple example, this is a hair strain from southern Siberian mummy. And what was done here was that this hair strain was sectioned into 1 centimeter pieces, and in each of these pieces, their carbon and nitrogen stable isotopes were measured. And if we focus on the nitrogen results, these go from about 9 to 12 per mil. And this shift here was interpreted as an increased consumption of freshwater fish. Now, to check if this was really the case, we decided to do radiocarbon measurements on these 2 hair sections because fish typically exhibits radiocarbon reservoir effect. So effectively, this higher nitrogen value should also have associated with it an older radiocarbon age. Now, these are the radiocarbon results, and as you can see, there is no significant difference between the two. So radiocarbon does not provide evidence for fish consumption. It doesn't mean there was no fish consumption, because the fish itself could have no reservoir effect, but it also opens the path for other possibilities, like a starvation signal, or simply having access to foods coming from different having access to foods having different isotopic baselines. Okay. Now, all of this data, isotopic data, can then be transferred to a Bayesian model to generate dietary estimates. And here I'll be talking about the fruits Bayesian mixing model. And this model has some features that set it apart from other mixing models. Fruits was designed specifically to reconstruct individual diets. And it has features that allow it to address in a simple way complex physiological mechanisms such as dietary routing. It also comes with a large variety of priors which are made available through a user-friendly interface, which means that it becomes very simple to incorporate into the model evidence coming from archaeological and historical sources. And of course, fruits is just one among many tools that should be used under this integrated approach, and here I would like to emphasize the future integration between fruits and ox skull, something that should become available next year. Now, as I said, the emphasis of fruits is the reconstruction of individual diets. And for example, in fruits, it's necessary to provide the uncertainties for the isotopic measurements made on the consumers. And the importance of this is illustrated in the results from this feeding experiment, in which mice were fed with different mice individuals were fed with the same diet, and yet the different tissues show isotopic ranges of about 1 per mil, which is larger than the measurement uncertainty. And so if this information is not given to the model, there is the risk of generating inaccurate dietary estimates. But fruits allows you to do much more, including addressing much more complex dietary models, include, for instance, routing mechanisms, different routing mechanisms. So you have here data from from one feeding experiment published in these two papers, in which groups of mice were fed with varying amounts of protein and lipids. So here you have 40% protein, here 5% protein, but 6% lipids and 41% lipids. The same amount of carbohydrates for both experiments. These are the isotopic signals of carbon for the bulk food and these for the protein. And the standard model assumes that all the signal in muscle or collagen of these consumers is being derived from the dietary protein. But when we look at the muscle signals for these consumers, there's a big difference between the two of about 3 per mil, which indicates that the lipids are also contributing signal to the consumer protein. Now I don't have time to go into this in detail, but here we made an attempt of using a scramble model. So the signal we used as input was a signal of the mixed food, and tried to estimate using fruits and different offsets, anywhere from -1 to 2 per mil between the food and the consumer. We tried to estimate what would be the diets of these consumers using this simple standard scramble model. Now, the correct true values are the red dots, and for one group of mice we have totally wrong diet. Here it's accurate, the estimate, and as we vary the diet to consumer offset, the reverse is obtained. So here we have an accurate diet description, and here it's completely wrong. And this is because we are not accounting for this complexity in terms of routing mechanisms. Now, I don't have time to go into this, but actually we can divide the consumer tissues into groups of amino acids, each one originating from these food fractions, carbohydrates, bulk, and essential amino acids. And we know what are the weighted contributions coming from these fractions, something that we can incorporate into fruits. And if we do that, we also need to, of course, take into account the isotopic signals of these food fractions. And if we do that, then we get accurate estimates. Now, all of this data coming from foods, coming from humans, coming from results from feeding experiments needs to be centralized so it can be handled in an efficient way. And that gave origin to the ISOMEMO initiative, which is a database of isotopic data, but also a platform bringing together different people and research groups interested in using isotopic data. Now, this initiative is following a bottom-up approach. There are about 90 different experts from the fields of archaeology, ecology, physiology, etc., providing feedback on the graphical interface, database design, data quality, etc. And of course, there will be plenty of applications coming out of this, but I would highlight in this context the possibility of establishing isotopic baselines, which are specific to a particular spot, to a specific region, and to a particular time period. And I should also add that ISOMEMO follows a partnership model by establishing partnerships with research groups in different institutions that already have their own isotope and radiocarbon databases. Okay, let's assume we have enough data to establish our isotopic baselines. We have data coming from these feeding experiments that we know how the offsets, how the signals are transferred from diet to consumers. We quantify their diets using a Bayesian approach, such as that involving fruits. Well, how can we combine the estimates generated using fruits with other forms of archaeological and historical data? And this is what I'm going to illustrate now. And I'm going to illustrate this by using data from publications involving isotopic analysis of humans, for which we have independent information on what were the diets of these humans. And I will start with first this publication by Coltrane et al. That came out in 2004, and it refers to the diets of Arctic foragers from the island of Silumut. So here we have a graveyard of Arctic foragers, and their bone remains were analyzed. Also analyzed were extensive faunal remains from the site. And here we have defined the two potential food sources and their isotopic signals. Those are terrestrial mammals and marine mammals. Now, these are the estimates generated by fruits, but we can improve the precision of these estimates by using information coming from the analysis of the faunal remains. This analysis established that the terrestrial mammals were far more consumed than marine mammals. And so we can provide this information to the model, and the fruits generated estimates become more precise. And we can actually compare these estimates with a study that was made of the estimated meat provided by each of these groups of mammals by looking also into the faunal remains. And these estimates are represented by the stars, and as you can see, there's an excellent agreement between fruits generated estimates and those generated from the study of the faunal remains. And now we move to medieval England, to the site of Timberhill, medieval graveyard in the city of Norwich, and data published by Bayliss et al. In 2004. So in terms of the isotopic baseline, isotopic signals were defined for 3 different food groups: terrestrial plants, terrestrial animals, and marine fish, using a variety of published data. These are the estimates generated by fruits for the caloric contributions coming from each of these food groups. But as you can see, there's a big imprecision on the estimates for terrestrial plants and terrestrial animals. But here we can use historic information that for the lower classes, the main source of calories were actually terrestrial plants. We can provide this information to the model, and now the estimates become much more precise. Now, I don't have time to go into details, but actually manorial records that describe food allotted to peasants working on these manors. This data has been collected, and the star symbols mark what are the estimates coming from these historical sources. And you can compare them with the estimate generated by fruits, and as you can see, there is an excellent agreement. So finally, I would like to talk about one case study where this integrated approach was actually applied. And that's the prehistoric graveyard site of Orstorf in the eastern part of Germany. Now, Orstorf site, the graveyard is actually located in an island within this lake Orstorf, which is located in this bigger lake district and about 1.5 kilometers away from this much bigger lake, Lake Schwerin. 3 food groups were defined: terrestrial plants, terrestrial animals, and freshwater fish. 4 dietary proxies were employed: carbon, sulfur, and nitrogen measured in collagen, and carbon isotopes measured in bioapatite. A variety of data was collected coming from modern sources, from literature, and from measured values, which will be available in the ISOMemo. And the diets were modeled using fruits and a complex rooted model, based on the results that we obtained from feeding experiments. Now, these are the estimates generated by fruits for the caloric contributions coming from each of these foods towards the group average, which we can summarize as follows. The majority of the caloric intake was coming from terrestrial plants. But if we focus only on the protein component of those diets, about 70% of those calories were coming from animal foods, and 45% was coming from fish. In terms of the amount of protein in the diet, compared with carbohydrates and lipids, this represented about 25% of all calories. This is a very high value. For example, modern populations and farming populations typically have below 15%. So this is much more characteristic of hunter-gatherers than farmers. And so one of the possible interpretations here in terms of models is that we could perhaps talk about a phenomenon of cultural resistance, in which both metaphorically and literally we have an island of individuals which adopt a lifestyle which is more characteristic of the Mesolithic, but they are surrounded by a sea of Neolithic of populations adopting mode of living which is more characteristic of the Neolithic. And to me this evokes this very classical example coming from Asterix and Obelix. The individual diets were also reconstructed, and here we have 6 men and 4 women. 3 of the women have amongst the highest fish consumptions. And so we can put forward the hypothesis that this dietary difference could also correspond to a labor division, with men hunting and women fishing. Of course, this is only a hypothesis. We would need additional evidence to corroborate this hypothesis. One of the women has one of the lowest fish consumption and one of the highest plant consumption. Could this actually be a farmer, an outsider, coming into this community? Now, these were individual graves, and each human was buried with tooth pendants, and these tooth pendants were of from terrestrial animals. They were radiocarbon dated, and also the humans were radiocarbon dated. And this was first published in the study by Olsen et al. In 2010. Well, we did additional radiocarbon measurements, and you can see here the results for animals, which fit nicely with the Middle Neolithic chronology. But the humans were considerably older than the animals, as you would expect, resulting from the consumption of freshwater fish, so they have a freshwater reservoir effect, a freshwater dietary radiocarbon reservoir effect. Now we can explore this to check the reliability of the estimates generated by fruits. But for that we also need to know the radiocarbon age of the fish. Now the environmental conditions today are similar to those in the past, so we could collect modern fish to determine this. But there were different possibilities, so we focused on the bigger lake Schwerin, which is located 1.5 kilometers from Lake Ostrov, and measure the radiocarbon reservoir effect for different species, and this is about 550 years. It is very different from the reservoir effect measured at Lake Ostrov. It is 1450 years. Now, what we have here is the age offset between the radiocarbon age measured in animals and that measured in the humans, represented by the red dots. The blue is that estimated by fruits, assuming a reservoir effect of 550 years corresponding to that of Lake Schwerin. As you can see, there is no good agreement. So Lake Schwerin is not the main source of fish for the populations at Ostrov. However, if we take as reference the reservoir effect at Lake Ostrov, then we have an excellent agreement. And so Lake Ostrov was the main source of fish for these individuals. Now, there's a clear outlier here, and this is probably a problem with the archaeology. And there's one individual for which the agreement is better with Lake Schwerin than that of Lake Ostrov. Could this be an outsider? And you can see here a first example of the use of radiocarbon as a food provenance proxy. And I hope that I've illustrated in this example how we can start building these models into the economy, society, and culture of these past populations under an integrated approach, and how truly powerful this integrated approach can be. Again, my apologies for not being physically present. I would like to thank you all for your attention, and if you have any question whatsoever, please do drop me an email. Thank you again.